Hello, everyone. My name is Nandini Jojode, and I'm a senior biology major at Adelphi University on the pre-medical track. My project is the characterization of a new player, the LSH3 protein, in a well-characterized hormone signaling pathway in Arabidopsis thaliana. Today, I will be talking to you all about a new gene that was discovered by one of my mentor students. This gene is called LSH3 and is found in plants, specifically in the cytokinin signal transduction pathway. My goals today are to show you what I have been working on for the past 2 years and hopefully educate you on the complex mechanism that exists in plants that allows them to grow in a way we experience every day. Plants are essential to our living, and one of the main ways plant growth is regulated is through a plant hormone called cytokinin. Plant hormones, just like human hormones such as insulin or estrogen, are signals that are sent to our cells. Our cells then relay the signal with the help of other proteins to target and amplify a response in different parts of the organism. So cytokinins are a group of plant hormones that regulate cell growth and differentiation in shoots and roots. So they also regulate processes such as embryogenesis, germination of seeds, nodule formation, and chloroplast development. So over here, we have a picture of adenine. Now, you might have heard of adenine. It's one of the 4 nitrogenous bases found in DNA. Cytokinins are called N6 adenine derivatives, and it gets this name because of the shape of cytokinin molecules, where they have the similar shape of adenine. However, they have different functional groups coming off of one of the nitrogens. So, a little bit about the cytokinin signal transduction pathway. So cytokinins are regulated in pathways in plants, and this one is known as the cytokinin signal transduction pathway, and is shown here. Cytokinin is the signaling molecule, and the rest of this image shows how different molecules are used to relay this message. So the first step in cytokinin signaling is for cytokinin to bind to a receptor on the cell membrane, and this receptor autophosphorylates itself and becomes activated. Now, all we need to understand here is that the receptor follows a cascade of events that leads us to the nucleus of a cell. In the nucleus, the signal can be transcripted with the help of transcription factors called Arabidopsis response regulators, also called ARR proteins. There are 2 types, type A and type B. Now, the signal is first received by the type B ARR proteins, and they regulate cell division. And this is the protein that we are kind of focused on today. Previous studies have shown the effect of 3 particular ARR genes, and these are the type B genes. It's numbers 1, 10, and 12, where all 3 of these subtypes have overlapping function and act similarly to one another. The study showed how when all 3 genes are removed from the plant, meaning they're knocked out or mutated, they show a very distinct phenotype of stunted growth or no growth. So we can see that right here, where it says ARR1, AR10, AR12, which means that these 3 genes are not present in this plant. And as you can tell, there's not really much of a plant here. Now, if you look at these 2 plants labeled wild type and AR10, AR12, they look quite similar phenotypically. The wild type is a regular Arabidopsis plant, while the second picture shows a double knockout where there's no AR10 or no AR12. However, ARR1 is still present. Now, like I just said before, since their functions are similar, the ARR1 gene is able to compensate for the loss of the AR10 and the AR12 gene. So Dr. Andreas Pfeiffer discovered the gene LSH3 since it binds to the gene ARR6 and represses or inhibits ARR function. This may be a new player in the cytokinin pathway. We realize that LSH3 is a part of the LSH family, which consists of 10 genes that all act very similar to one another, just like the ARR genes work. So since it would be harder to isolate a single protein and knock it out or mutate it in the plant, we decided to overexpress the gene to understand its function. So my hypothesis is that LSH3 overexpression will mimic the phenotype of an ARR1, ARR10, ARR12 triple mutant. So I came to this conclusion since LSH3 represses ARR. Now we know that ARR is needed for cell division. So if ARR is not present and two of them are already knocked out, such as like in this plant, when there's LSH3 overexpression, meaning there's more levels of LSH3, it means that ARR1 won't be able to function properly. So with that being said, I decided to go on with my project. So my approach to this was to overexpress LSH3 in Arabidopsis thaliana. So this is a model organism since it's easy and inexpensive to grow. It has a small genome and has a short generation time and can produce a lot of seeds. So as I grow these plants, I also noted down the phenotypic characteristics of the different plant lines to find any correlations between LSH3 expression levels in mutant plants versus wild-type plants. So wild-type plants are just regular Arabidopsis plants with nothing special about it. There's no mutations or anything in it. My final goal is to relate this back to the cytokinin signal pathway and hopefully figure out LSH3's function in the future. So this first table right here shows the different plant lines I worked with. So I had different control and experimental groups. So line 1, which says wild type, is shown by this picture right here. Now, the wild type plant is just the regular plant and it has no modifications. Line 2 is the AR10, AR12 plant, which is shown right here. This plant has modifications where the AR10 and AR12 genes are not present in the plant. Line 3 shows a triple mutant, just like the one that we saw right here. And this one shows the same exact thing, but you can see that there's some little plants growing here. But it's not as noticeable as the AR10, AR12 knockout or the wild type. So this is where things get interesting. So we decided to have a AR10, AR12 knockout and also increase LSH3 levels. So when we did this, we came up with this plant and this one. So both of these lines have the LSH3 overexpression, and they are the AR10, AR12 mutated plants. So this is what I was referring to in my hypothesis. Now, if you look closely at line number 8, which you might not be able to tell, but there is growth inhibition because the area of the leaves is not the same as the wild type. It's actually a lot smaller. Also, it also has stunted growth than the wild type. In line 9 over here, we have the area of the leaves is smaller, but it grew in the same height as the wild type plant. So since phenotypically we did see something that was peculiar or interesting, we decided to go further ahead and do a Western blot. A Western blot is a scientific technique that is used to determine the amount of protein in a sample. The Western blot on the left, which is shown right here, shows the plants that have LSH3 overexpression. So as you can see here, we have plant number 8, which is seen in this well, and plant number 9, which is seen in this well. And both of them are showing bands for LSH3. So since we got this, we decided to also have a control experiment to make sure that we weren't missing anything. So the Western blot on the right is a Western blot using tubulin, which is a very abundant protein in plants. So we used this blot to make sure there was equal loading of protein in each gel, and this served as a control. So over here for lines 1 through 7, we don't see anything in the Western blot. However, here for tubulin, we see it. So this means that protein was added into the gel plates, but none of the protein was LSH3. So these results were very promising. The blocked bands of LSH3 overexpression are indicative that the stunted plant growth may be due to LSH3's ability to inhibit the ARR1 gene, which inhibits normal cytokinin signal transduction pathway. However, since these plants did not fully develop, they did not reproduce further seeds to be analyzed for more testing. So I then made new plant lines with LSH3 overexpression. So my project currently is to grow these new plants and characterize them for LSH3 overexpression to make sure that we mutated the plants correctly. So our first step is to sterilize our seeds in order to avoid contamination and to grow it on a sucrose petri dish, as shown here. Once the seedlings have formed, I transfer them into pots with fresh soil and watch the plants grow and note any peculiar phenotypes. Finally, I perform a Western blot to analyze LSH3 function. So this is done for every line, and it's done over a couple of generations to make sure that we have LSH3 expression in every seed that we get. So you might be wondering, why would I spend my last 2 years solely taking care of plants? Well, cytokinin is very versatile. It's an essential hormone in all plants, and understanding its pathway can help formulate new technologies for human benefit, such as production of biofuels and protection of plants against pests like insects. Some of the broader implications of performing a project like this is not just for my curiosity and plant biologist curiosity, but understanding a complex pathway like this helps us understand plants better. In the world that we live in today, where there's overpopulation and finite resources, geneticists around the world have been trying to find solutions to impossible problems, such as world hunger and biological resource depletion. With climate change creating imbalances, it's becoming more inevitable than before to protect our food resources. The effect of climate change will have an effect on the plant's efficiency to produce food and the material we need from it. And if we understand the cytokinin signaling better, we will be able to manipulate plants in a way to provide food security in the future. Thank you so much for listening in. And finally, I would like to thank Dean Dinan and the Honors College at Adelphi University for providing me with this wonderful opportunity. Thank you to Dr. Pfeiffer for discovering the role of LSH3 in this pathway. Thank you to Dr. Alan Schoenfeld for his constant help and feedback on lab techniques. And I'd especially like to thank Dr. Lawrence Hobby and Dr. Alexander Heil for being my mentors in exploring this fascinating subject. Thank you all, and I hope you have a wonderful rest of your day.